Hello, my name is Erica Anderson, and I'm the Section Chief of the Cytogenetics and Genomic Microarray Laboratories at ARUP Labs, and an Associate Professor in Clinical Pathology at the University of Utah. I'm also a member of the ACMG ClinGen CNV Interpretation Guidelines Committee, and today I'll be providing my perspectives as a laboratory director on implementation of the new technical standards. But before I do so, I wanted to provide an update on behalf of the ClinGen Dosage Sensitivity Curation Working Group, in which I'm a co-lead on the subgroup working on recurrent CNVs. So we recognize that there's a lack of consensus in the community on the classification and reporting practices surrounding reduced penetrance CNVs, and there are several ongoing efforts to assist in resolving these issues. Given the enthusiasm surrounding this topic, I wanted to provide a brief update on work being done by the Dosage Sensitivity Working Group to update the curation process and reviews for the recurrent CNVs. So over the past 2 years, our group has been working on a metric for evidence scoring specific to recurrent CNVs. This metric is new, but evolved out of existing curation processes for genomic regions, and includes various evidence types that also reside in the clinical CNV interpretation metrics, including evaluation of evidence relating to genes within the CNV, such as gene count and HI predictors up at the top. In the middle, case and cohort level data, including specificity of the phenotype and inheritance patterns. And at the bottom, case control and population level data, which have established scores based on p-values, effect size, and CNV frequency. Most recently, we've been evaluating so-called subclinical phenotypes, which include measurable deficits or differences in neurocognitive function and related features that have been examined in individuals identified from well-phenotyped control populations. Approximately 100 unique CNVs corresponding to over 50 recurrent regions have been incorporated into the project thus far. Currently, we have a number of regions evaluated that are in the dosage sensitivity 3 category, and we've worked our way up from the dosage sensitivity unlikely category into examining the more challenging and sometimes controversial CNVs scored in the 1 and 2 categories. On the right, comparing this new metric to our historical scoring for these regions, we've generally had good concordance in these methods, though some CNVs have and will undergo scoring changes. And so we'll keep the community updated on our website, which is now hosted at theclinicalgenome.org. So these are our goals for this year, which in addition to what I've mentioned, include updating scoring and reviews for regions such as the 15q11 BP1 BP2 deletion and the 16p13.11 dupe. And we should have updates coming for these regions this winter. In addition to curation scoring, we've also been discussing forming a VSEP for the recurrent CNVs that will provide expert level clinical interpretations for the recurrent CNVs in ClinVar. I also wanted to mention that in February of this year, the ClinGen Low Penetrance Risk Allele Working Group published a framework intended to describe all clinically significant genetic variants, regardless of population frequency or variant type, including the multigenic CNVs. In our lab, we've started to adopt this new classification system by adding a reduced penetrance qualifier for recurrent CNVs with established clinical phenotype association and penetrance estimates. But we have not yet adopted a term such as risk allele to describe these variants, and we'll wait for consensus to be reached on discriminating between these two categories. Next, I'll share our perspectives on implementation of the new CNV interpretation technical standards from ARUP, where this work is still in progress. And so I'll start out with a little information about us. My focus here is going to be on implementing the metrics for evaluation of results from genomic microarray testing. And one of the inherent limitations to this is that breakpoint determination at the exon level is typically not possible in order to determine whether, for example, reading frame may be disrupted in areas of the metrics where this information is assessed. ARUP is a reference laboratory. We run constitutional genomic microarray testing for post and prenatal testing as well as pregnancy loss. And so the indications do vary, and the extent of clinical information that's provided to us may be somewhat limited. This affects our ability to establish presence of all potential matching phenotypes to those provided in cases from the literature, as well as to review results from other clinical and genetic testing that may have been performed. In addition, as is the case for most array testing, parental testing or trio analysis is not typically performed up front to assess inheritance status for CNVs. We do offer parental testing following a report for a proband in the prenatal setting for VUS, where this practice is standard of care and can provide some information in the absence of the ability to look for postnatally encountered phenotypes. But where we've actually found the metrics to be quite useful is when our scoring is near classification boundaries. For example, a large VUS that's determined, if determined to be de novo, may be upgraded to likely pathogenic. And so as we work to implement the new technical standards, we've made changes to our reports and reporting practices to align with new recommendations. For one, we've moved away from solely using more generic language in our report headers to including a 5-tier classification section up at the top of our reports. This has been somewhat challenging to implement for certain variant types, such as deletions conferring autosomal recessive disease risk and the recurrent CNVs. So I wanted to share our approach and how we manage these. So for deletions involving recessive disease genes, we've created different report templates based on classification and the presence of other genes. For example, for pathogenic and likely pathogenic deletions that are disease-causing in a dominant manner but also happen to contain recessive genes, we mention this risk is also conferred in the interpretive section of the report, but don't typically highlight or describe individual genes unless there's a phenotypic match we feel compelled to mention. Same goes for VUS that are both multigenic and reportable regardless of whether they happen to contain a recessive gene. Here we use a VUS heading and mention AR genes are also in the deleted interval. For VUS that contain additional genes but would not otherwise be reported, we focus the report on AR risk. So these are generally smaller deletions with fewer genes of uncertain significance and tend to lean towards the likely benign classification due to, for example, observation of similar variants in control databases. And then, of course, for deletion limited to an AR gene, we also focus the report on this observation. And as I mentioned earlier, for recurrent CNVs, we've started to use the generic reduced penetrance qualifier to reports for CNVs for which penetrance estimates are both available and established. And so there should be a reasonable sample size and resultant confidence interval around such estimates in order to use them. For this, we've been using a threshold of 60%, below which we'll tack on the reduced penetrance qualifier, and studies such as the Eichler and Signature Labs case control and inheritance comparisons, as well as newer large-scale studies that have focused on individual recurrent CNVs, have really been key resources for determining this information. And so, speaking on behalf of the Dosage Sensitivity Working Group, we intend to have this information updated also on the public site. So in terms of our experience using the metrics thus far, and questions submitted to us ahead of this webinar, is the new framework for CNV classification increasing the number of VUS and LB classifications? In our experience, yes, but appropriately in many cases. We've found so far that by starting at uncertain, we're becoming more accurate in our classifications and in documenting our rationale for downgrading CNVs, which helps us move away from traditional classification approaches such as prioritizing CNV size over gene content. Implementing a more standardized approach should provide better accuracy both within and across labs, is necessary to improve the quality of data and data shared on a large scale, and will also facilitate better retrospective data review and reevaluations of previously reported CNVs, which is becoming more commonplace, at least at our institution. Another way we're able to manage starting from uncertain is by leveraging both our internal database and platform-specific common CNV tracks in order to downgrade CNVs to likely benign and benign. We're still looking at exact thresholds for this, but our aim is to establish at what frequency we can reasonably assume a particular CNV is benign or is likely benign as a primary method for downgrading. Another question is whether the new standards are increasing our reported CNVs. And the answer to this is not yet. We do track our VUS rates and have not seen those increase there. And we do not report likely benign CNVs. And while we generally report VUS, we still do use size criteria, which are particularly relevant for small duplications that are otherwise uncompelling to report and follow. And we'll be taking a look at the recent Mayo study to determine whether further refinement for our duplication reporting criteria is needed. So this is our roadmap to fully implementing the new interpretation metrics. We're currently refining our clinical staff's roles in evaluating and documenting information from different steps of the metrics, and developing a workflow that's outlined in the figure at the bottom of the slide. So at ARUP, our array technologists review the data and provide a preliminary CNV classification, then draft reports which come to the directors for review. Some of these variants are routed either from the bench or directors for investigation by variant scientists who will work through the metrics and provide, where appropriate, an updated classification. And then we also enroll members from our genetic counseling team to help create report templates and ensure the classification provided matches the information we're communicating, and includes the appropriate phenotype and recommendations for follow-up. In order to implement these changes, though, we're working to standardize CNV classification terminologies, interpretive comments, and documentation. And then we're also looking at methods to automate certain aspects of the CNV analysis workflows, such as determining gene counts and overlap with known benign and pathogenic genes and regions, which is another advantage of implementing these approaches. In terms of documentation, as part of this project, one of our fellows, Jian Zhao, created two versions of investigation templates, one for CNVs with multiple genes and another for gene-level CNVs. So here I'm showing you the multigenic CNV template, which includes gene content information at the top, overlap with internal and external gene and region annotations in the middle, and summarize information from the detailed evaluation of section 4 of the metrics at the bottom. And so here's an example of a recent approximately 1 megabase duplication in 11q25 that we investigated, the boundaries of which is highlighted in blue in the UCSC Genome Browser snapshot, provided as a link out from the first page of our investigation template. So here it was noted that within this duplication, the region in orange appears to be commonly duplicated, but is too dissimilar in size and gene content to be used to downgrade this duplication outright. You can see that while this region does not appear to represent common variation, there are also only 9 protein coding genes here, and with limited data on gene disease associations, especially for duplications. So as I mentioned, we typically start by looking in our internal database, and here you can see that the duplicated region displayed at the top doesn't really overlap any dupes similar in size to the one that we're investigating, though a few encompassed by this region were not previously reported. And as the lit review is the most variable aspect of multigenic CNV investigations, We also include documentation of search terms used to query and identify potential reports similar to our duplication from searches in PubMed and the internet. In this case, with no additional data, the duplication remained a VUS. So here's another snapshot from a different multi-genic CNV investigation where we determined the protein coding gene count and modified this as instructed in the technical standards. In this case, there were 2 gene families shown on the right-hand side that needed to be accounted for appropriately within this region. And this is just being shown as a reminder. And then finally, I'm sharing our investigation template for gene-level CNV evaluation, which incorporates information specific to exon and gene-level evaluation, such as the transcript isoform reference and breakpoint analysis, if feasible. So I'd like to acknowledge everyone from the Genomic Microarray Laboratory at ARUP, including our clinical team of medical directors, fellows, lab staff, scientists, and genetic counselors, which include members of the team working on the array workflow project. And I thank you all for your attention.